Hey guys, my name is Gino. I'll be giving a brief overview on the topic of enzymes and their importance in biological systems. In this presentation, I'll be going over enzyme functions, activation energy, and enzyme inhibition. So let's get started. Enzymes. What are they? Enzymes are biological catalysts that are important because of their ability to accelerate or catalyze chemical reactions that would otherwise take place in a non-useful timescale. In other words, magnitudes of years. So I think the best way to illustrate this or to demonstrate this ability is when we look at an uncatalyzed reaction, any uncatalyzed reaction. But in this specific example, considering the combustion reaction of sucrose here, or table sugar. So in this balanced chemical reaction here, we have sucrose here reacting with molecular oxygen, producing carbon dioxide and water. And if you measure the standard free energy change of this combustion reaction, we get negative 6,054 kilojoules here. And if you've taken a intro to physics or intro to chemistry class, you would recognize that because of this delta G0 being negative, it's actually thermodynamically favorable because it's exergonic, right? It's releasing energy into the surrounding. Yet despite this condition here, this reaction actually occurs in a time scale of years. However, in the presence of a catalyst, sucrose oxidation actually occurs in seconds, and this is evident when you eat sucrose, and because we're humans and we're able to metabolize this carbohydrate, right, and use it as for fuel, utilize it for energy. So that goes on to our next point here, that practically every chemical reaction in a cell occurs at a significant rate because of the presence of enzymes. And without these enzymes, you can probably tell that we wouldn't probably live that long, right? Because chemical reactions won't occur, and products aren't made, and cells will die because these products are required for the cell to survive, right? And so, how do they actually do this? Right, so enzymes provide an environment in which a given chemical reaction can occur more rapidly and favorably. And what I'm talking about in this environment is this is in reference to the active site of the enzyme, which is the region of the enzyme denoted here, in which the chemical reaction actually takes place in, right? So the substrate or the reactant actually comes and binds to this region of the enzyme, and this is where we have the enzyme substrate complex, and this is where the chemical reactions actually occur. We'll go on to that later. But within this active site of the enzyme, the reactants or substrate are correctly oriented such that productive collisions occur and the reaction proceeds. And these productive collisions occur because the environment is suitable for that reaction to occur. There's a lot of ins and outs to that statement, but essentially what I can tell you is that this environment is very specific. It has a very specific acid or basic environment, or it could have the presence of cofactors or non-protein helpers that help the reaction to occur. And of course, without these cofactors, the enzyme wouldn't be functional, and the reaction wouldn't proceed, and products won't be formed. And just to continue on with this figure here, again we have the enzyme here, which is really simplified. It's a really simplified drawing, but if you look at images on Google or whatnot, you will see that these enzymes are very complicated in structure. They're mainly composed of amino acids, but of course we have some exceptions to that. And within them they have their various cofactors and other prosthetic groups that are not included in this figure, but are included in those other figures in Google. But just for the sake of this presentation, here we have a generic enzyme. Within it, again, the active site where the substrate will bind to, and this enzyme substrate complex forms. Complex here's disappeared. Sorry. And as the reaction here takes place, we have our bonds that are broken, or bonds being formed. Things are being rearranged within within the active site, and then it spits out your products here, which are denoted in red. Again, this is very generic, very general, and there's a lot that goes into it. But if you look at a physical perspective, we need to consider the reaction's activation energy, or the amount of energy required for a chemical reaction to occur. And this is best illustrated when we look at a reaction diagram, a reaction coordinate. And it's extremely important to understand this concept because it gives us insight on what a chemical reaction requires in order to occur. All right. So let's break this down a little bit to understand it a little bit better. Right. So on the y-axis here we have the free energy here, or if you want to be more specific, the Gibbs free energy here, denoted as G. And on the x-axis here we have the reaction progress here from reactants A into product B. So if we start off here early in the reaction, we only have reactants present in the mixture or whatever vessel that we have here, or in the cell. Let's say this is in the cell. And as we go through time, we see here that we need to actually invest some energy to overcome this barrier in order to form products B. And this energy invested in the reaction is known as the energy of activation, or the activation energy here. Again, in the definition, the amount of energy required for this chemical reaction to occur. And this is denoted as here, as you can see these dots here, which are kind of more or less estimated. I'm sorry. And again here, the free energy, or I'm sorry, the energy of activation here is denoted as the delta G double dagger superscript here. But in common textbooks that it is denoted as is actually EA, energy of E subscript A, energy of activation here. And I also put this subscript here down over here to denote that that this reaction is actually an uncatalyzed version. And so if you look at the blue line here, the blue plot here, we see that this energy here is about the same starting. Then you still even catalyzed, still need to invest some energy here in order to form, again, products B. And again, this energy invested is known as the activation energy, but in this time it's the catalyzed activation energy. Denoted here also is the transition state. This area of the diagram is extremely important because it is this point in the reaction where the chemical species or the reactants are extremely unstable, right? They can, if enough energy is invested, they can fall down and then go towards products. But if not enough energy is invested or the chemical conditions aren't right, it can go back to reactants A. Again, this also happens in terms of this transition state is also lowered here in terms of the catalyzed version of the reaction. And so this reaction coordinate, again, is really important because it illustrates the fact that every chemical reaction needs some energy invested in order to form its products B. And relevant to that also is the change in free energy, delta G here. This delta G here, as you can tell, is not changing despite having a catalyst in the reaction mixture. And that's really important to recognize, because if the delta G changes, this whole reaction will change from an exergonic reaction, which you may have guessed here, being negative relative, this energy level being more negative relative to this initial starting point, initial energy level. So the same token, you can actually mimic this in an endergonic reaction, right, where delta G is actually positive, right? And that's an exercise that I kind of want to give you guys. So what happens if the delta G is actually positive? How is the reaction going to be affected in that case? And of course, you can always Google it, but it's good practice to really test your understanding of what's going on in this reaction coordinate. So two important takeaways here that I kind of want to give you guys is that the enzymes, again, do not change a reaction's change in free energy, right? I mentioned that if you change this free energy, like you change the whole reaction, this exergonic cannot become an endergonic, right? But again, instead they lower the energy of the transition state. As you can tell here, you can also tell that this transition state energy level is much higher than the catalyzed transition state level here, right? The second point is also that the activation energy of a non-catalyzed reaction is far greater than the energy of activation of a catalyzed reaction, which is consistent with the definitions I've been giving you guys, right? And so that's really important to go about. I know I've been going that pretty fast, but again, it's a really important concept to understand. in terms of looking at this in a physical perspective. So the third topic I kind of want to briefly go over is enzyme inhibition, right? And these inhibition plays, again, a vital role in the metabolic regulation in cells. And of course, this is important because if enzymes are in control, they would continuously catalyze reactions and continuously use reactants. And this might not necessarily be good for the cell because it's pumping out, or it's using resources, right? And it's pumping out like products that it doesn't need, right? And so these products are wasted, energy is wasted, and the cell won't survive without regulation. So inhibition comes in two types. We have the competitive inhibition and we have the non-competitive inhibition. And here in, Here in competitive inhibition, we have this is the case when an inhibitor competes with the intended substrate for the active site of an enzyme. When I'm talking about an inhibitor, I'm talking about a chemical species, any chemical species that is, again, structurally similar to that of the intended substrate. And you would realize, or you would recognize that, a really central theme in biology is that structure dictates function, right? And so if you have a chemical species or a reactant that is very chemically similar in structure to the substrate, you can guess that that species will actually bind, that is capable of binding to the enzyme. And that's kind of what happens in terms of a competitive inhibition, right? So when the inhibitor occupies the active site, the intended substrate can no longer bind to that enzyme because it's occupied. And so the catalysis does not occur. So here I have a nicer diagram of what actually happens, or what is really generic and what happens in this case. So normally here we have, again, the generic enzyme and the generic active site of the enzyme. Here we have a substrate, any chemical species, and it can bind to it, and this binding forms an enzyme substrate complex, which will then catalyze the reaction, which will form the products denoted in this square and circle products. However, when you look at if you have an inhibitor here, and I really like this diagram because it kind of looks like this shape here, but it really isn't. It looks like that shape, but because it is structurally similar to the substrate, when it binds to the enzyme, no product is formed. And you can also notice that we have a reversible equilibrium expression here. And this is a little bit more complicated, but yes, we also have a reversible and a non-reversible binding of inhibitors. And depending on what the inhibitor is, that will obviously dictate what type of inhibition is occurring. And in some cases, even these permanent inhibitors are your poisons, right? Your cyanides, which are just generic poisons or neurotoxins and whatnot that permanently can disable the enzyme and cause various disorders and diseases. So the second type of inhibition also is non-competitive inhibition. This is the case when the inhibitor binds to the enzyme at a site other than the active site, right? This is a site or a location of the enzyme that is not the active site, right? And such binding induces a conformational change to the enzyme, which can directly inhibit the intended substrate from binding at the active site or reducing catalytic activity. And that's a mouthful. But what I mean by conformational change is that when the inhibitor binds to this inhibitor site, this can actually induce a change in the enzyme structure such that when the intended substrate attempts to bind to the enzyme, it won't be able to, and pointing towards this diagram here. I'm sorry. So again, it's best illustrated in this. Here you have the normal case where the inhibitor site is not bound, and the enzyme is, or I'm sorry, the substrate is capable of binding to this the active site, and you'll be able to have catalytic activity and therefore products. However, in the case of an inhibitor, and the inhibitor goes into the equation, it will block the active site, or I'm sorry, the inhibitor site, which will therefore change the conformation or the structure of the enzyme, and therefore the substrate is no longer able to bind to it, and therefore little or no product formation occurs. Again, this is very general. There's a lot of ins and outs of it. You can actually model this mathematically, but I think I'm going a little bit over time here. But again, enzymes are extremely important, and this basic understanding is important when you look at very real world examples. And I kind of want to, in light of the respiratory illnesses that are kind of going around, I want to give an example that is not really the coronavirus, but really similar to it. Again, that's the influenza virus, right? And so this example I'm giving you is an example of competitive inhibition, and the drug used is Tamiflu. As you may know, Tamiflu is a drug used to treat influenza infections or flu infections, and essentially its mechanism of action is actually that the Tamiflu, the drug Tamiflu, is a competitive inhibitor that blocks an essential enzyme of the virus or influenza from doing its activity. Right. So let's kind of look at its mechanism a little bit more carefully here. So the enzyme is known as neuraminidase, right? And its job or its role in the virus life cycle is to cleave off a sugar link from the host cell so that the virus is able to spread in the bloodstream and therefore infect other cells here. So here I kind of took a snapshot of my textbook here that describes a part of that virus or the flu um, life cycle. And of course, there's a lot of steps here, but this is the point of the life cycle that we actually kind of have, as us humans, have the ability to kind of prevent, right? By means of utilizing Tamiflu, the inhibitor, which blocks neuraminidase activity here, and specifically, this cleaves the sialic acid link of the host glycoprotein, which is essentially just a carbohydrate bound to a receptor here. So here we have in this case, if you can see my highlighter over here, we can see that the virus here buds out, and neuraminidase actually cleaves this portion here of the sialic acid receptor and therefore releases the virion, right? However, if in the presence of the inhibitor or in the presence of Tamiflu, this prevents the neuraminidase from cleaving the acid receptor. So the virus will continue to remain attached to the cell, right, and won't be able to spread because it's not cleaved. And that's just one of many examples of the importance of enzymes and inhibition and a working knowledge of it. But that's my time, and I think and I hope you guys learned something from it and stay safe. Thank you.